Okay, so welcome back, everyone, to this session on cell division. So we are going to cover the cell cycle, an outline of the cell cycle. We are then going to talk about mitosis and cytokinesis separately. And we're also going to describe the stages in meiosis 1 and 2. And we're going to also discuss how it introduces genetic variation. And we're going to contrast it with mitosis, which is a really typical question as well. So first, our outline of the cell cycle. The main phases of the cell cycle, the purpose, first of all, sorry, of the cell cycle. Oh, sorry, I appear to have gone back in a slide. Sorry, just give me a sec. The purpose of the cell cycle is to facilitate growth and replacing dead or damaged cells in multicellular organisms. I'm just going to do a very quick link to the chapter about DNA, and I'm just going to mention that the whole reason why cell division is possible in the first place is because DNA is the only molecule that can replicate itself, and that really facilitates cells being able to divide and daughter cells being able to inherit a full set of the genetic material. DNA can replicate. There are three main phases to our cell cycle. The first phase here in red is interphase. And we can immediately see that the cell spends most of its time in interphase. We then have mitosis. Let me just label that in blue just to keep with it. So mitosis is here. And finally, cytokinesis is a separate stage after mitosis. Now, an important term distinction. The word cell division is not synonymous to the word mitosis. So cell division, I'm just going to jot that here. Cell division is not synonymous to mitosis because cell division includes cytokinesis. The cell division is made up of mitosis plus cytokinesis. Actually, mitosis specifically means nuclear division. So for a cell to divide, it spends a significant portion of time to separate the nuclei and then to separate the rest of the cell, the cytoplasm and the rest of the cell. There are some important cellular functions that are happening during interphase. And we could broadly describe interphase as when the cell is not dividing. And importantly, when DNA is replicated. So interphase is basically separated into three main stages. And the first stage is called G1, stands for gap 1. And this is when the cell will synthesize proteins and organelles and physically grow. It will then include what we call the S phase, which is when DNA replicates itself. And then it will include growth, gap 2 phase. And there's going to be a little bit more growth happening in gap 2, but there will also be a checkpoint that will check for mutations. And if the cell has, if there are any mistakes, any wrong bases, any mutations accumulating during interphase, the cell will not proceed to mitosis so that these mutations are not inherited to the daughter cells. Note that for AQA and EdXLA is not, you don't need to know about the checkpoints, but it's worth remembering them. They relate very much to cancer and how tumors develop, so we're not going to go into them now, but you need to know about them. So, as we said, mitosis phase is the nuclear division of the cell. Cytokinesis is the full splitting of the cell into two daughter cells. Let's look at a typical example question that kind of tests our knowledge and understanding of the entire cell cycle and not just mitosis. So in an investigation, a student observed a stained section of mammalian liver tissue. They counted how many cells could be seen in each stage of the cell cycle, and the data is shown below. Use the information provided to calculate the percentage of the cell cycle taken up by nuclear division. So context, right? The data. So this entire thing is my context. Command, calculation. I'm just trying to, right? So calculate. End direction. We have to assess so taken up by nuclear division. So we've got to translate that in our head into which one of those stages represent nuclear division, and then calculate the percentage for 3 marks. So I'll give you just under 3 minutes. 1 minute per mark is what you're recommended to spend in exams. See if you can have a go before I give you the answer at how you would approach this. It's a little bit challenging, but see if you can have a go. Or at least consider what strategy you'd use for this question before we go through it together. So, first of all, let's really kind of connect the context and the direction of the question to how we will approach it. I'm being told, just to kind of remind you, I'm being told that I need to calculate the percentage of the cell cycle taken up by nuclear division. So what constitutes nuclear division? In other words, mitosis. Well, it's the phases, excluding interphase. So it's prophase, metaphase, anaphase, telophase, which are the 4 phases that make up nuclear division. So I will not include interphase in my calculations of how much time cells spend in nuclear division. That's not to say that I'm not going to utilize this number at all, because since I am calculating a percentage, it means what? The number of cells doing what I'm told, nuclear division, as a proportion of all of the cells. So what will be actually my first step here will be to calculate the total number of cells. So if I add these numbers up, it comes up to 500 cells in total. So 500 cells in total were observed. And now we want to see what proportion, what subset of those was in nuclear division. Just to sort of super quickly clarify that the rationale behind this question is that the more cells I observe in one phase, that suggests to me directly that this will be a longer phase, because if the longer a phase takes, the more chances I'm going to catch cells at that phase. So this question could equally ask me, what is the longest phase of the cell cycle? Then I'd say interphase. Or what is the longest phase of nuclear division? Well, then I'd say telophase, because that's where most of the cells are found. So my total number of cells are 500. These are the number of cells at nuclear division. And so if I now add up these phases, if I add up prophase, metaphase, anaphase, and telophase, and again, I would suggest, guys, make it clear. oops, sorry, make your working clear, as this is worth 3 marks. If I add up these numbers, they add up to 90. So 90 cells in total out of 500 are in nuclear division. So what do? I want to calculate the proportion of that, or the percentage. And so I'll do 90 over 500 times 100 to get the percentage, which works out to 18% cells. I hope that's clear. Now, let's move on to the actual mitosis and cytokinesis, so breaking down each of the intermediate steps here. As always, the specification points for reference, make sure you're super familiar with them. So let's first of all then define mitosis. So, and what are the features about mitosis that we have to know? So mitosis is actually just one cell division. You'll see that this contrasts later with meiosis. And it produces genetically identical daughter cells. That also contrasts with meiosis in a minute, and will be really clear. So this is my parent cell, and it divides to form 2 daughter cells. Daughter cells that are also genetically identical. It is used to form somatic cells. Quick reminder of the term somatic. It basically means every cell in the body apart from gametes. So it produces somatic cells and not gametes. And why is that important? Growth and repair of multicellular organisms, as well as asexual reproduction in unicellular eukaryotes. The type of cell division by which prokaryotes divide is called fission, not mitosis. So if you see the word mitosis, mitosis specifically refers to the particular behavior of chromosomes and what the cell does. You will not say that bacteria divide by mitosis. So let's go and see the phases one by one of mitosis. First phase is called prophase. And what are the events that are happening at prophase? First of all, DNA condenses to form visible chromosomes. How does that happen? By DNA supercoiling. So DNA supercoils and it condenses to form visible chromosomes. Prior to that, DNA still exists in the nucleus as chromatin, meaning DNA wrapped around histone proteins. If you haven't done that yet, don't worry, you will. But it's in the form of it just kind of looks like a loose sort of string-like structure in the nucleus, not as visible rods that are chromosomes. The second thing that happens is that the nuclear envelope disintegrates. That's essential if we're going to split the chromosomes. And so here we can see no nuclear envelope. We can see that the chromosomes are condensed, invisible. What's another thing that happens? The centrioles move to the pole. So these here are the centrioles. If you don't remember what they are, revise your chapter on cell structure. Centrioles are those 2 little organelles that duplicate and they move to opposite poles of the cell at the start of cell division, at the start of mitosis. And the centrioles organize the mitotic fibers. So we'll be able to see the mitotic fibers. Just do them in a different color so you guys can see them better. You'll be able to see the mitotic fibers like that, sort of extending across the nucleus. So the mitotic fibers emanate, we say, from the centrioles. What happens in metaphase? In metaphase is when the chromosomes are actually going to line up along the equator of the cell. And that's when the mitotic fibers, or the mitotic spindle, I'm just going to say that it can be called spindle as well instead of fibers. Just be aware of that. Mitotic spindle oh, sorry, spindle fibers. Sorry, my bad. I meant mitotic spindle instead of spindle fibers. Can be also called that. It exists from prophase, but at metaphase, it will actually attach itself to the chromosomes. Which part of the chromosomes? Specifically the centromere, which is this little central part here where sister chromatids are held together. This is my centromere. These are my spindle fibers, or mitotic spindle. These are, again, the centrioles. The chromosomes then start to be pulled along the two opposite poles of the cell by the spindle fibers. These spindle fibers have the ability to contract and pull each sister chromatid to opposite ends of the cell. And so that's where it starts to prepare them to actually separate. Which then brings us to the next stage, which is anaphase, when the actual physical separation of the sister chromatids is taking place and they're being pulled to the opposite poles of the cell. The separation is aided by motor proteins that are able to contract and physically pull against the chromatids. This is an active process. It requires ATP. So you can sometimes see sort of little visible mitochondria kind of arranging themselves around the spindle fibers so that they can provide ATP. The chromosomes will have this kind of characteristic appearance. The chromatids pulled apart, and they will actually look like they're being dragged through the cell. Beware that many questions will actually show you microscope images of the phases of cell division, and it's a really good idea to sort of train your eye to familiarize yourself. But this will be one of the ways that you'll know you're looking at anaphase versus looking at metaphase when the chromatids are aligned along the equator of the cell. So each pole of the cell will end up with identical and full sets of chromosomes. That's the whole purpose of first lining up the chromosomes along the equator and then making sure that each daughter cell only receives one sister chromatid each, so that they've got both a full set and an identical set. Since the function of mitosis, part of the function is to create growth or to replace dead or damaged cells, they have to be genetically identical. Otherwise, they wouldn't be replacements of the cell that was lost. Finally, telophase, the nucleus begins to reappear. It's beginning to form again here. So here is my nuclear envelope reforming. And I, of course, have two new nuclei. So, and they're all part in the same cytoplasm. So the cell appears to have two nuclei for this brief moment in time. The chromosomes begin to decondense. So you can see they don't have that distinctive chromosome appearance. It looks like a sort of loose fiber. Decondense. And the spindle fiber also begins to disintegrate. It's not needed anymore. The chromosomes length and uncurl, meaning that they can no longer be seen under a microscope. You may be able to see some kind of diffuse staining, but you won't be able to identify rods. And of course, as we said, the nuclear envelope reappears around the chromosomes, around the chromatin. Let's have a look at an exemplar question here. So after fertilization, a zygote rapidly divides to produce more cells. Each cell cycle may be as short as 30 minutes and only involves DNA replication and mitotic divisions. State the type of cell division involved in this process and suggest why the produced cells become smaller after each cycle. So take a moment to answer that. I'd suggest that you do try to write out an answer, because you can then compare it to the model answer that we're going to give here. And that's going to be really good for you to see exactly where you go wrong. It's going to help you to kind of pinpoint your areas that you need to work on. You could plan the answer in your head, but writing it down is better. Okay. Let's look at the answer together. So the context is the whole first sentence. So the context tells us that we are dealing with cell divisions, rapid cell divisions to produce somatic cells. And so our command is state. So we, in terms of stating the type of cell division, it's just the one, but also suggest an explanation. So that's going to be the rest of the 2 marks. So 1 mark will go for state, 2 marks will go for the explanation. What is my direction? Why each cell division gets smaller. So that is what I need to be able to justify in my suggest part. So this is my direction here. So let's see how we're going to break down this answer. So first of all, let's identify the type of cell division. It's kind of already given to me also in the question here, but it's basically mitosis, since it's somatic cells being produced. So that's 1 mark. I'm just going to briefly state that sometimes this particular type of cell division, where there isn't much growth between cell divisions, it can also sometimes be called a cleavage. It's not in your syllabus explicitly, but just if you see it, that it doesn't throw you off, just have it in the back of your mind, this term. So they divide through mitosis, and usually cells will spend more time in growth 1 and growth 2 phases, where they will undergo protein synthesis and make organelles, which is basically what growth is, isn't it? Making more organelles and synthesizing proteins, translating proteins. Without these, so this is now we are suggesting why these cells are smaller, become smaller after each cell cycle. They spend less time. I was told that because I was told that the cell cycle might be as short as 30 minutes. That was a clue. And it only involves DNA replication and mitotic divisions. So which stage of the cell cycle is missing? Growth 1 and growth 2. Without growth 1 and growth 2, no organelles. Without these stages, the amount of cytoplasm and the number of organelles must be divided between the daughter cells, so they effectively almost half with each cell division, so the size reduces. Let's now move to cytokinesis, which is not a part of mitosis. The cyto means that, like, we're the chapter mitosis and we're looking at cytokinesis, but of course cytokinesis is a separate stage, and it will involve the separation of the rest of the cell components, the cytoplasm and the cell membrane, into 2 identical, of course, because we're talking about mitosis, daughter cells. It happens slightly differently in plants and animals. In animal cells, where there's no cell wall and the plasma membrane is it's elastic, it's non-rigid, it's fluid, right? As the fluid mosaic model tells me. So it will actually fold, and there are 2 dents. Let me just, I hope you can see that. There are 2 little dents here formed. For those of you that want a bit of extra detail, these dents are called the cleavage furrow, but don't feel like you must memorize that, just in case you want to. And it will basically constrict around the whole cell, and it will form 2 separate daughter cells. Plant cells do have a cell wall, which is rigid, it's inflexible. And what will happen, there's not going to be any constriction of the membrane, no folding, there will actually be a gradual formation of cell wall, so cellulose, and then let me just do it in a separate color so that it's obvious, and cell membrane beginning to form. So it's kind of like building a fence, imagine. So I'm going to have plasma membrane, and this structure holds the end plate here, which is made of cellulose. So the end plate will eventually become the new cell wall that separates the two daughter cells. And that will form two separate plant cells. Let's have a look at a typical question, which is also a calculation question. So E. coli can divide as often as once every 20 minutes, as can most bacteria. So that is my context here. Context in green. Assuming that one E. coli cell weighs 1.5 picograms. Quick unit explanation. A picogram is 1,000 times smaller than a nanogram, which is 1,000 times smaller than a microgram. Don't worry too much because I'm actually told the unit conversion here. So calculate the amount of time taken for 24 picograms of E. coli to grow from a single cell. So my context, we said, is the data here. The command word is, of course, to calculate. Do it in red. Calculate. And my direction is what I need to actually calculate here. So take a moment again. I'm going to give you a couple of minutes to see how you would approach that. It's challenging to have a think. Even if you don't solve the whole thing, plan it in your head. So think about, first of all, how you will convert the information into number of cells. How are you going to find the number of cell divisions required since you're only given the amount of time for one cell division? What's the kind of strategy that you want here? Let's break down our approach to this question. So, how much time is taken for 24 picograms of E. coli to grow? First thing I need to do is to convert that into number of cells. Okay, because I'm only being given the amount of time that it takes for one cell to become two, to divide. So I have to convert the information given to me in picograms in terms of numbers of cells so that I can actually work with, so that I can utilize this piece of evidence here, the 20 minutes for one division. So how are we going to convert those picograms into numbers of cells? So I'm just going to write down like our strategy here. Calculate number of cells that are corresponding to 24 picograms. I'm being told that each one cell weighs, this is where you've got to be utilizing every piece of evidence that you're given. So how will I utilize this information that one cell weighs 1.5 picograms? So I will do 24 picograms divided by 1.5, which is the weight of one cell. And that gives me the number 16. Obviously, this is picograms. This is picograms. The units cancel each other out, the top and bottom of the fraction. So that gives me the number 16. So 24 picograms correspond to 16 cells. Okay, now I'm getting somewhere because now I know that I need to go from one cell to 16 cells. So next I need to figure out how many rounds of cell division will this take to go from 1 to 16 cells? How many cell divisions? So you can do sort of like sketch that out in your head. So one cell becomes 2, that's one cell division. 2 cells become 4, that's a second cell division. 4 cells become 8, that's a third cell division. And 8 cells become 16, because that's what I worked out here. That's a fourth cell division. So I need 4 divisions to take place. Okay, now the solution is nigh, because I know that each one cell division takes 20 minutes. Just quickly, you can write it out like that if that kind of like makes more sense to you, to count basically the number of cell divisions, or if the numbers were different and it would be impractical to write out all the cell divisions, you could also use the formula 2 to the power, 2 to the power of n. Sorry, that's meant to be an n, not an h. N, where n is the number of cells. Sorry, number of cells is 2 to the power of n, where n is the number of cell divisions. So since I know that each cell division takes 20 minutes, and I know that to go from 1 cell to 16 cells, I need 4 cell divisions, then my total time, because I need the total time taken is of course going to be 4 divisions times 20 minutes, which is 80 minutes. You can write minutes like that, or just write out minutes just to be sure. Okay, that's it for mitosis. We are now going to move into meiosis. And again, make sure that you're super familiar with your spec. So meiosis, and you're going to see that each of these points now basically contrast it to mitosis. So it involves 2 cell divisions as opposed to 1, and it creates haploid instead of diploid, gametes instead of somatic cells. Don't worry, we're going to do a much more sort of focused comparison between mitosis and meiosis in a minute. The first meiosis occurs after chromosome duplication. So this parent cell is still going to go through interphase, which means it will still go through S phase. So its chromosomes will be duplicated. Meiosis 1, crucially, and this is important because you do get tested often on which chromosomes separate in which phases of the cell division. It separates homologous chromosomes into different cells. So this is my result of meiosis 1. And here are my homologous chromosomes separated. Chromosomes separated. Just really quickly, homologous chromosomes are chromosomes that have been inherited from a different parent, and that's why they've got different colors here, black and yellow. So, for instance, if this is chromosome 1, this is chromosome 1 paternal from the father, and this is chromosome 1 again, but inherited from the mother. When meiosis begins in the reproductive organs like the ovary and testes, it starts from a diploid cell, and every diploid cell contains 2 sets of chromosomes. Humans have 2 sets of 23 chromosomes, meaning I've got chromosome 1 from my mom, chromosome 1 from the father, chromosome 2 from the mother, chromosome 2 from the father, and so on. So here's now my homologous chromosomes separated. And then meiosis 2 finally will actually now go ahead and separate these sister chromatids from each other. And so this is meiosis 1, this is meiosis 2. And these are now my sister chromatids that are separated. Definitely make sure that your chromatids, definitely make sure that you're really comfortable with the terms homologous chromosomes versus sister chromatids and what they mean. Sister chromatids are genetically identical to each other and they're held together by the centromere. Homologous chromosomes are not identical to each other because they contain different alleles that were inherited. They've got the same genes in the same locations, but they've got different alleles because they were inherited by different parents. So the phases of both meiotic divisions, meiosis 1 and meiosis 2, are the same. It's prophase, metaphase, anaphase, telophase. And in terms of the general events, they have a lot of similarities with the phases of mitosis. So in prophase 1, chromosomes supercoil or condense, nuclear envelope breaks down, and spindles form, same as mitosis. What's the difference, though? That at the end of prophase 1, that homologous chromosomes are being separated in meiosis. In mitosis, only sister chromatids are separated, ever. In meiosis 1, it's homologous chromosomes that are separated from each other. So yes, all the mechanics are the same, nuclear envelope, chromosome condensation, mitotic spindle forming, but the types of chromosomes that are separated is what's different. And a really important event takes place at the end of prophase 1, which is crossing over, which is when sections of DNA are exchanged between homologous chromosomes, between the chromatids of homologous chromosomes. And so for AQA and Edexcel, you don't need to know the stages of meiosis. The reason is because they're very similar to the stages of mitosis. So if you learn the stages of mitosis, you're fine, and you just need to know how they differ from meiosis, basically. Meiosis 1, metaphase. So in metaphase, the chromosomes, if you can see here in this diagram that we've got regions of alternating like light blue and dark blue. So for instance, this chromosome here with the light blue might have been maternal chromosome, and this chromosome in the dark blue might have been paternal chromosome. And you can see that they've exchanged little portions of DNA between them. They now line up at the equator, and the spindle fibers are bound to this diagram should show you know that the dotted line is going directly onto the centromere. And this is where we have an important event called independent assortment or segregation. Both are correct. What does this refer to? So as we said, homologous chromosomes are not genetically identical to each other. They come from different parents. They contain different alleles. Which daughter cell, the position of them in the equator, determines which daughter cell is going to inherit which chromosome. Meaning what? These could have been flipped. So paternal chromosome could be going to this cell, and maternal chromosome could be going to this cell. Equally, this chromosome could be going on this side, this chromosome could be going on that side. This process, importantly, is random. It's a random process. And it introduces genetic variation. Why? Because the daughter cells are not going to be genetically identical, since they will each have a random mixture of paternal or maternal chromosomes. What then happens during anaphase and telophase? The chromosomes from each homologous pair are actually physically going to be pulled to the opposite poles of the cell, much like in mitosis. And then finally, at telophase, the chromosomes are going to start to uncoil, decondense, and the the nuclear envelope begins to reform around those two new nuclei. So here we have anaphase 1, and here we have telophase 1. The two nuclei that are temporarily formed each now contain one set each of the chromosomes. So we're going to see this point again in a minute. At this stage, at the end of meiosis 1, these daughter cells that are going to form after cytokinesis are already haploid because they only contain a single copy of the homologous chromosomes. Let's have a quick look at a question before we move on to cytokinesis. Before we move on to the meiosis 2. Mitosis and meiosis are two types of cell division, which produce somatic cells and gametes, respectively, in mammals. Respectively. State another difference between the cells formed by mitosis and meiosis. So one difference is that we were told that they contain, let's look at our context first. Our context is right here, comparing meiosis and mitosis. Our command word is state, no justification. It's only worth 1 mark as well. So brief, keeping it brief here. And my direction is to focus on the differences of cells produced. So what does that mean? You cannot mention just any difference between mitosis and meiosis, of which there are many. And this is why this question is only worth 1 mark. Otherwise, a comparison between mitosis and meiosis would be worth more marks, of course. But here we can only talk about the cells produced. So have a quick minute to get to think about the answer before I say it. So one difference is what type of cells are made, somatic versus gamete. And what's the other difference between those two cells? Well, somatic cells are diploid and gametes are haploid. So mitosis produces diploid cells, which all somatic cells are, whereas meiosis produces haploid cells. Just because it's 1 mark doesn't mean that you shouldn't write like a whole coherent sentence, right? Cells. Okay. Let's now move to the second meiotic division, which again is made up of prophase, metaphase, anaphase, and telophase. So, as we said, meiosis 1, before we start with meiosis 2, produces 2 haploid cells because there are no homologous chromosomes anymore to speak of. Maternal chromosome 1 from paternal chromosome 1 have gone to the 2 daughter cells. Doesn't matter which way around they went, but there are no homologous chromosomes anymore. So technically, if we define diploid and haploid with the respect of whether we've got sets of chromosomes in there, we are now at a haploid state. It doesn't matter that we've got sister chromatids still, because sister chromatids, so these are two haploid nuclei, or cells actually, even though they have sister chromatids still, which of course are going to be separated now in the second meiotic division, because sister chromatids are genetically identical to each other. So they're not considered homologous chromosomes to each other. And we define haploid or diploid by whether we've got homologous chromosomes or not. So now in prophase 2, what is going to happen? DNA will supercoil, nucleus disintegrates, spindle fibers form. Same as in any beginning of meiosis or cell division. Metaphase 2, the chromosomes are lined up at the equator of the cell. Now, while sister chromatids in mitosis are, generally speaking, genetically identical, sister chromatids here are not identical to each other. Why? Due to crossing over. So just remember this diagram here, which just shows it a bit more clearly, that the normally this sister chromatid, and this sister chromatid would be genetically identical, but because of crossing over, and crossing over, remember, introduces genetic variation because it creates new combinations of alleles. We're going to see that in a minute. This sister chromatid to this sister chromatid is not genetically identical. So why is that relevant for meiosis 2, for metaphase of meiosis 2? Because this basically introduces yet another round of independent assortment. So during anaphase, when the genetically different chromatids are separated to opposite poles, this leads to a second round of independent assortment or segregation, meaning what? Which sister chromatid ends up in which daughter cell means that I will have two nuclei that are, of course, not genetically identical. So the first independent assortment is meiosis 1, homologous chromosomes are separated. The second one is when those sister chromatids are separated, and the reason they're not genetically identical is because of crossing over. So we have now 4 genetically different and haploid cells where also the sister chromatids have gotten separated. Let's have a look at an exemplar question. Meiosis is used by organisms to produce gametes, such as sperm and egg cells. Explain why meiosis requires 2 stages of cell division. So, what is our context that we are talking about? Meiosis. That first sentence here. Oops. Context. Command. Explain. So, justify your choice in the context of this question. And then finally, direction. We have to describe what happens in these 2 stages of meiosis 1 and 2. Take a minute to structure and possibly write the question. Let's look at the answer together and break it down. So meiosis requires 2 stages in order to, 1, half the chromosome number. So this is one bit of crucial information where 1 mark goes to. What's the other thing that meiosis does? It separates the homologous pairs of chromosomes as well as sister chromatids. So that's my 2nd mark. So those are basically, I'm describing the jobs of meiosis, right? It's required to separate homologous chromosomes and then sister chromatids, effectively halving the chromosome number. Let's look at crossing over. So we're going to talk about specifically how meiosis introduces genetic variation into sexually reproducing organisms and how that's an advantage of sexual reproduction over asexual reproduction. So what does crossing over do? We phrase it as, so first of all, it allows alleles to be swapped between homologous chromosomes. So as we said, homologous chromosomes are not genetically identical. They will have the same loci, meaning the same gene. So for example, in this position here, let me just use a different color. Sorry, guys, because you probably won't be able to see that really well. And it's important that you can write down the annotations if you need to. So I'm just going to go with red here. In this position, there will be a gene that controls, for instance, eye color, right? And I'll have the same gene in the same location, whether I am looking at the, let's say this is the paternal chromosome, let's say this is the maternal chromosome. So this individual has a diploid cell before meiosis takes place. So here we are before meiosis, before prophase 1, because it's during prophase 1, at the end of prophase 1, where crossing over takes place. So this is the alleles that are found on this chromosome. So this person is, for instance, homozygous for capital A, and they're also homozygous for another gene that codes for a different trait, gene B, that will be found in this particular location. So they are homozygous for uppercase B. The maternal chromosome, again, as we said, it's going to contain the same genes at the same locations, but different alleles because it came from another person. It came from a mother. So say that this person was homozygous here for lowercase b, and then they were also homozygous for lowercase a in this part of the chromosome. When these sections of chromosome swap between the other, so what happens is that the homologous chromosomes, they line up. They actually line up, or we can say pair up as well, because line up might make you think about metaphase, where they line up along the equator, whereas homologous chromosomes actually pair up. This is an important detail. They pair up during prophase 1, where they may not necessarily be exactly at the equator yet. Prophase 1. And at that stage, pieces of chromosomes literally intertwine, they twist around each other, and they physically exchange portions of chromosomes. So it causes these sections of chromatids to break off, and they're swapped to the homologous chromosome. Yes, they only involve one of the 2 sister chromatids, whichever one is closer to the other, but the sections that they exchange are between homologous chromosomes. So don't say that they exchange between sister chromatids. If this sister chromatid exchange a piece with this sister chromatid, I would never know because they're genetically identical. The sister chromatids are still genetically identical at this stage because they haven't undergone crossing over yet. So what can I see here? So this sister chromatid is going to be the same as before. It's going to be capital A, capital B down here. However, here it has exchanged. Sorry, I should have drawn the A a little bit lower. I just noticed. But it has exchanged that piece of chromosome. So now it's going to be lowercase a here, and it's going to be lowercase b down here. And on this section, it's going to have uppercase A here. Sorry, that was meant to be red. Uppercase A here, and lowercase a here as it had before. And it's going to have now here uppercase B, and it's going to have lowercase b as before here. So I say, and that's a really good phrasing to use, that it creates new, just write it here, new combinations of alleles, because capital A used to go with capital B and lower a used to go with lower b, and now I've got a lower a with lower b here, and so they've exchanged places. So it creates new combinations of alleles. This phrasing basically comes up in a lot of mark schemes, so it's a good one to use. They will still have the same genes, so same genes at the same positions of the chromosome, but different alleles. What about independent assortment or segregation, and how does that help with, or contribute rather, sorry, to genetic variation? First of all, how do we define it? It's the random distribution of homologous chromosomes along the cell equator in metaphase 1, which affects which chromosomes, and therefore alleles, are going to end up in different daughter cells. Again, try to copy that phrasing, maybe not word for word, but making sure that you include all of the keywords that you need. And this random distribution of sister chromatids in metaphase 2, which also will affect which alleles end up in daughter cells. Let's kind of like try to sketch that out so that we can understand what we mean. So again, let's say that we're starting off with a diploid cell here, 2n, and this diploid cell has, we're not going to draw 23 chromosomes that humans contain, but let's just see it through the example of 2 chromosomes. So here's 1 chromosome. Let me just do red and blue so it's super clear. So this diploid cell will contain 1 maternal, for instance, chromosome 1, and another chromosome 2. So this is chromosome 1, chromosome 2 from maternal, and it will also contain their homologous chromosomes. So paternal chromosome 1 and paternal chromosome 2. When the first meiotic division takes place, how could these chromosomes separate from each other? Well, one daughter cell could inherit, for instance, both maternal chromosomes. So we go chromosome 1, maternal, chromosome 2, maternal. And the other cell could inherit both paternal chromosomes. 1 paternal, 2 paternal. However, they could also be the other way around. So I could have and that will happen if maternal chromosome 1 is on this side of the equator compared to the other. What if those chromosomes line up differently in the equator of metaphase? Well, so I could then have here paternal chromosome on this side, and the other paternal chromosome, chromosome number 2, can go on the other side. And equally, I can have my maternal chromosome 1 on this side and my maternal chromosome 2 on this side. So now how will these chromosomes separate into daughter cells? Well, now I'm going to have maternal chromosome 1 here with paternal chromosome 2 here. And now I'm going to have paternal chromosome 1 here, and I'm going to have maternal chromosome 2 here. So you can see that the daughter cells are not genetically identical. So the number of different egg or sperm that can be produced from a meiotic division are always going to be different. There's going to be genetic variation of them. I just want to make a really quick note that pay really close attention to whether the question is asking you, how does meiosis contribute to genetic variation? In which case you will talk about the random distribution of homologous chromosomes or independent assortment of homologous chromosomes and the independent assortment of sister chromatids in metaphase 2. If the question asks, how does sexual reproduction as a whole contribute to genetic variation, then you will also mention the fact that it's the random fertilization of gametes as well that contributes to genetic variation. Let's see a typical question on meiosis, and particularly on the how meiosis contributes to genetic variation. So crossing over and independent assortment are critical for introducing genetic diversity during meiosis. Describe the process of crossing over for 3 marks. So again, let's break this down. What is my context? It's meiosis and genetic diversity, the relationship between the two. What is my command word? It's my command word is describe, yes. But I'm not going to go into too much depth. I'm just going to basically describe or outline in a way the stages of crossing over and how it comes about. And finally, my direction is that I'm only focusing on crossing over and not independent assortment. So not independent assortment. So take a minute, it's 3 marks. So take a couple of minutes to see. Oops, sorry. I'm so sorry. Switched over to the answer page too quickly. Take a couple of minutes to outline the stages of crossing over. Let's have a look at the answer together and break it down and comment on it. So one thing that we can say about crossing over is when it occurs, right? So it occurs in meiosis 1. And it allows alleles to be swapped between homologous chromosomes. That's also kind of like close to how we would define crossing over, right? So that's 1 mark. The homologous chromosomes line up and wrap around each other. So now we're describing the actual process, which causes bits of each chromatid. You can say sections if you don't want to say bits. Sections of each chromatid to break off and exchange with each other. That's a 2nd mark. Effectively swapping alleles. That's my 2nd mark. The points where crossing over occurs are called chiasmata. That's something that we didn't explicitly cover. The chi, for those of you that are familiar with the chi-square test, chi just means x in Greek. So they call them chiasmata because they literally look like they form an X as they're swapping the bits around, the sections around. Let's go to the next bit. So an actual sort of comparison between mitosis and meiosis, very common question. Whenever you've got two similar processes in biology, you just know there's going to be a compare and contrast question in there. So it's important that we can, and whenever you are making any kind of compare and contrast questions, it's really good to be able to separate out what aspects of them you're comparing. So with respect to how many divisions there are. It's one cell division and makes two daughter cells. Two cell divisions makes four daughter cells. The next aspect is whether there is recombination. What do we mean here by recombination? We mean recombining DNA sources that are not identical. Daughter cells end up being genetically identical. So you can see here that they're identical. They contain the same copy number of chromosomes and an equal mix of maternal versus paternal chromosomes. Whereas little cells are haploid and they only have one set of chromosomes. Bear in mind that this idea of haploid versus diploid, as we saw in that question, can also be somatic versus germ cells or gamete cells. Not germ cells, gamete cells. Let's have a look at an example of one of these extended response questions comparing mitosis and meiosis. I'm going to give you a few minutes to try and come up with the answer yourself before we look at the answer together. But let's first do our strategy of breaking down the question. Explain how meiosis increases genetic variation and compare the products of meiosis and mitosis. This is actually a pretty generous phrasing, I would say, because sometimes they might literally just say compare and contrast meiosis and mitosis. Whereas here we're kind of like told that you also need to make sure to include in your answer the products of mitosis and meiosis. We would do so anyway, even if we weren't told, because that's part of what comparing mitosis and meiosis is. But now we're told explicitly. 1 mark is given for the quality of written communication. So again, what is my context? It's this first bit here. It's, sorry, so sorry. I was so used to, see, my bad. I jumped ahead. My context here is that I am dealing with mitosis and meiosis. So those are the divisions I'm going to compare. My command word is explain. And explain with some detail as well, since it's an extended response question. And finally, my direction is that since I'm comparing, let me just change color here. Since I am comparing the products, I need to include comparative language, comparative adjectives or language. While, whereas, things like that. Oh, sorry, I did it again. So sorry. Take about sort of 3 minutes to 4 minutes to have a go at the answer. Okay. Let's have a look at the breakdown. So out of those 3 marks, so 3 probably, 3 will go towards explaining how meiosis contributes to genetic variation. About 2 of the marks are going to contrast the products of each one of mitosis and meiosis. And then finally, of course, 1 mark goes for the quality of the QWC. Sometimes these are abbreviated in the mark scheme for examiners. Quality of written communication. So let's see the answer together. Mitosis produces. So we're starting by comparing the products. Mitosis produces 2 genetically identical diploid daughter cells. So genetically identical, let me just use red. Genetically identical diploid daughter cells, that's 1 mark because those are the products. While meiosis produces 4 genetically distinct haploid daughter cells, that's my 2nd mark. So those are my 2 marks for comparing the products. Variation meiosis can be caused by recombination between homologous chromosomes, i.e. crossing over during prophase, that's 1 mark. You get 1 mark for stating that it's between homologous chromosomes, and you get another mark for stating where, when, and where it happens during prophase 1. Or by the independent assortment of homologous chromosomes, which occurs both in metaphase 1, so independent assortment of homologous chromosomes is 1 mark, which occurs in metaphase 1, so homologous chromosomes in metaphase 1 and sister chromatids in metaphase 2. It's really important that you distinguish. Don't just say homologous chromosomes and sister chromatids. Define which cell division they take place in. And that's another mark here.